Today we are going to discuss cell injury, adaptations and cell death, which is the second chapter of Robbin's and one of the most fundamental topics in pathology. This area is extremely important not only because it forms the basis of almost every disease but also because it is a highly tested subject in examinations. Cells are usually resilient, capable of adapting to normal stresses. The adaptation is up to some extent. When the cell is unable to adapt, cell injury occurs. The cell injury occurs due to injurious stimuli like ischemia and hypoxia, toxins, infectious agents, immunological reactions, genetic abnormalities, nutritional imbalances, and physical agents like trauma, high or low temperatures, radiation, electric shock, and sudden changes in atmospheric pressure. The cell injury can be reversible or irreversible. When the injurious stimulus or the stress is mild and transient, the injury is reversible and the cell can be converted to the normal homeostasis. When the injurious stimulus is severe and progressive, the injury becomes irreversible, leading to cell death either from necrosis or apoptosis. As we discussed earlier, ischemia and hypoxia is an injurious stimulus. Ischemia means reduced blood supply and hypoxia means reduced oxygen due to reduced blood supply to that particular organ. Due to low oxygen, mitochondrial function is lost because mitochondria needs oxygen for its oxidative phosphorylation which produce ATP. Due to low oxygen, mitochondrial dysfunction occurs, which is the first change in cell injury, and reduced oxidative phosphorylation ultimately leads to reduced ATP production. ATP is the energy currency of the cell. Reduced ATP leads to low sodium potassium ATPase activity, which leads to sodium accumulation inside the cell. Sodium inside the cell drags more water into the cell, leading to cellular swelling. It is called hydrophic changes, which is the first morphological change in reversible cell injury. With cellular swelling, organelles also get swells. Example, endoplasmic reticular swelling. And the microvilli structure is flattened and ultimately get lost, and the cytoplasmic blebs also be formed. And there occurs myelin figures. Myelin figures are cells and organelles swells. There occurs some amount of cell membrane damage. It leads to myelin figures, which is seen as this kind of structure in microscopy. It is mainly made of phospholipids and it is like an lamellated structure. And with reduced ATP production, the aerobic respiration is reduced and the anaerobic respiration takes over the energy production and produce lactic acid as an product, which leads to increased lactic acids leads to lactic acidosis, and acidosis denature chromatin and clumping of nuclear chromatin can be seen. It is called as nuclear changes. And due to reduced ATP and the swelling of endoplasmic reticulum, the attached ribosomes of the endoplasmic reticulum get detached and decreased protein synthesis occurs. So in reversible cell injury, the first change of cell injury is mitochondrial dysfunction, which ultimately leads to reduced ATP production, and it leads to cellular swelling or hydrophic changes, endoplasmic reticulum swelling, loss of microvilli, cytoplasmic blebs, and myelin figures. And lactic acidosis leads to clumping of nuclear chromatin, which is called nuclear changes, and endoplasmic reticulum swelling leads to detachment of ribosomes and reduced protein synthesis. We discussed that cell injury are of two types, which is reversible and irreversible. When the injurious stimulus is severe and progressive, there occurs irreversible cell injury or cell death. When the membrane is severely damaged, it causes increased calcium influx into the cell. Calcium is an enzyme activator which activates phospholipases, proteases, and nucleases. Phospholipases attack cell membrane phospholipids and mitochondrial membrane and get damaged. Calcium going into the mitochondria binds with the inner membrane and form large amorphous flocculent densities, which is the hallmark of irreversible cell injury. And in irreversible cell injury also myelin figures can be seen. And due to activation of proteases, cytoskeletal proteins get damaged, leading to cytoskeleton damage, and nucleases, nuclear DNA gets damaged. DNA damage are three types, like the first, condensation of nucleus, which is called pyknosis, and fragmentation called karyorrhexis, and dissolution called karyolysis. Condensation, pyknosis; fragmentation, karyorrhexis; and dissolution or lysis, karyolysis. Irreversible cell injury or cell death can occur either due to necrosis or apoptosis. Necrosis is always pathological and it is a passive process, which means there is no need of ATP, and the most common type is coagulative necrosis. Necrosis means accidental cell death. Apoptosis means it can be either pathological or physiological. Apoptosis means programmed cell death, which is an active process, in turn needs ATP for its activity. First, let's discuss about apoptosis. There are two pathways of apoptosis, which is extrinsic pathway and intrinsic pathway. Extrinsic pathway begins with Fas ligand binding to death receptor or CD95 receptor. This binding signals the cells to die. The receptors trimerize inside the cell and it is called activation of the receptor. Activates FAD. FAD means Fas associated death domain. This FAD activates procaspase 8 and 10 to caspase 8 and 10, which is called initiator caspases. And in intrinsic pathway, there are stress sensors inside the mitochondria like BIM, BID, BAD, NOXA and PUMA. And apoptotic factors are there like BAX, BCL, XS and P53, and anti-apoptotic factors like BCL2, MCL1 and BCL-XL. The stress to the cell is recognized by the stress sensors, or sensed by the stress sensors release cytochrome C into the cytoplasm. This is due to the stress sensors activates pro-apoptotic factors and inhibits anti-apoptotic factors. BCL2 here is the anti-apoptotic factor which gets inhibited by the stress sensors. Due to the inhibition of BCL2, the cytochrome C is released into the cytoplasm, and this cytochrome C is bound with APAF1, which is apoptosis activating factor 1, binds with cytochrome C, forms apoptosome. This apoptosome converts procaspase 9 into caspase 9, which is also an initiator caspase. This 10-9 and 8, 9, 10 caspases are initiator caspases. They activate executionary caspases 3, 6 and 7, which in turn activate phospholipases, proteases and endonucleases, which makes apoptotic bodies from the pathogen. These apoptotic bodies have eat me signals or signals given by phosphatidylserine to macrophages, and macrophage engulf these apoptotic bodies and forms phagosomes, and these phagosomes attach with lysosomes and form phagolysosome complex, and ultimately the cell get lysed. This is the whole process of apoptosis.